for the people that couldn't attend, so they can take a look later. So again, welcome to the COP, the community of practice. For the ones that are just joining for the first time, this is a group that likes to share on topics related to breeding scheme optimization, germplasm management, and other topics that are relevant for the Crops to End Hunger initiative on increasing genetic gains and variety turnover. So, this is a safe community. We like to hear the comments, feedback, without necessarily attacking people. We want to hear from you as well. So if you want to present in this community of practice, please just send us the topics that you would like to hear. It's always difficult actually to find, people send the comments on topics, but they don't volunteer to present. So please, I encourage you to volunteer and present something nice from your program. I think that I have talked to many of the CG breeders and they actually are doing interesting stuff. So it would be nice if one of these days we have some of the forages breeders or bean breeders or maize breeders presenting some of their nice stuff. Their cassava breeders are also doing great things. So again, I encourage you. For today, we only have one topic, but I think something very interesting, especially for the clonally propagated crops. It's kind of an advanced topic related to genomic prediction. And actually for this year, we want to go back to the basics on the multi-environmental trials and indices, etc. But if you remember, the last 3 sessions, we've been trying to wrap up some of the guidelines and recommendations for implementation of genomic prediction for increasing accuracy, intensity, and also cycle time. And I think we want to just close that that series by bringing something that is, I think, relatively new and that also kind of shakes some of the foundations of how breeding is actually done. I hope Christian refers to that part when we always talk about breeding value, and I think we have to expand this horizon a bit more. So I will leave you with Christian Werner, and he will present on genomic prediction, cross performance in the clonally propagated crops. So Christian, do you want to take from here? I think this will be very interesting, and we will still save some time at the end for comments, questions, and just discussion in general, in case something was not clear or you would like to elaborate or contribute to the discussion. Christian.
Thank you, Eduardo. Hi, everyone. Can you hear me well?
I can hear you well. What about the rest? Is there someone that can confirm that?
I can hear you.
I can hear you.
Excellent, great. So Eduardo already gave a brief introduction. I'm going to talk about a project that I worked on during my time at the Roslin Institute when breeders from Driscoll in the UK approached us for support with the implementation of genomic selection in their strawberry breeding program. And therefore I will use strawberries as an example, but our results, they will hold true for basically all kinds of clonally propagated crops or species. And that's why we thought this is probably a very interesting and important topic to share today in one of the COP meetings. The talk will probably take about 30 minutes. Feel free to ask questions during the presentation already if something is unclear, but please keep in mind that we only have 1 hour, so let's please save the longer discussions for the end. And I will start with the key message, so you already know and you know what to expect from this presentation. So what we found is that let's call it conventional genomic selection approach, which is based on genomic estimated breeding values, is not suitable to select parents in clonally propagated crops. I will explain why this is the case, and therefore we advocate what we call genomic prediction of cross performance to identify the best parental combinations in a clonal breeding program. but let's start from scratch. You're all very experienced, and I'm sure you know that, but just to make sure we're on the same page, we have a baseline. What does a common clonal breeding program look like in general? So there's a schematic representation here. We normally start with crossing relatively heterozygous parents, and doing this induces sexual recombination, and we create a population of seedlings. And in terms of that strawberry breeding program, we always had somewhere between 10 to 20,000 seedlings created from maybe 100 crosses. So quite a large number of seedlings here, created by sexual recombination. And after that, we select the best individuals and clonally propagate them, though asexual propagation. This gives us the first stage of clones, also called A-clones. And from that stage on, we do what we do in every kind of breeding program. We try to select the best individuals, and we reduce the number of genotypes, but we increase the replication they're tested in across years, across different stages, and across environments. And when we have better estimates of the performance of those individuals, at some stage, we also start selecting new parents to induce a new generation. And if all that works out well, at the end, we will end up hopefully with a new variety. And we used this representation of a strawberry or a clonal breeding program now to figure out what might be a good strategy to include genomic selection here. So first thing we came up with is kind of a baseline scenario, something what we thought is relatively simple to implement. We used the basic key structure and assumed that our stage one, our first generation of clones, could be used as a training population. And of course, because these individuals are also tested in later stages, we produce more phenotypic information, which also then later will feed into that model, into the training population that we use to train a genomic selection model. So this has a couple of advantages already. First is that when we create a training population and use genomic selection, our individual of selection does not, or our individual of evaluation is not really the single clone anymore. The allele becomes what we make our selection on. And also the individuals are not replicated at that stage. The alleles and the combinations of alleles within individuals are replicated within generations, within the different stages across years, and we get a good estimate of the effect of the alleles or the allelic combinations. That means we can increase the selection accuracy in that early stage. And when we can do that, we can also make earlier selections, reduce the generation interval, one of the biggest advantages of genomic selection. And we also use genomic selection here to move or to advance the best predicted individuals to later stages. Eduardo explained that in one of the earlier sessions, that one of the big advantages of genomic selection is actually when we do have only very limited information, which means unreplicated or very few replication, then extracting information from the genomics relationship matrix, from other stages, from replications, from other years, massively helps to increase the selection accuracy, the foundation to reduce the generation interval. As I said, we used that as a baseline scenario, but we also realized that there are a couple of issues in clonal breeding programs which probably require another approach. And I will try to talk about them again based on the example of strawberries. So when we look at the seedlings are sexually propagated. They're normally unreplicated. Sometimes they're replicated, but only very, very limited replications possible. In strawberry, they're grown in these matted floors, in these matted rows on the ground, on the soil. And selection is in general based on a very rough and general visual assessment among these huge numbers of individuals. The clonally propagated individuals are asexually propagated. They might be unreplicated in earlier stages, but we get replications in later stages. And they're grown in these single pots on tabletop, on vertigated, so highly controlled tabletop systems. And we have a trait-specific assessment with increased accuracy. So there are a couple of issues with that step from the seedlings to the clones. One of the issues is you find in literature that apparently sexually propagated and asexually propagated individuals, although it's the same genotype, they might differ in their performance, they might differ in their morphology. I don't know why that is the case, but that is apparently observed, and that makes selection a problem. And this problem is aggravated by the fact that these individuals are grown in completely different environments. So we probably have strong G/E effects here. And these two issues result in the fact that we probably can't make reliable selections here in the seedlings when we're actually interested in clonal performance, asexually replicated, and in a different environment. On top of that, we have that very general visual assessment. So eventually we end up with a very, probably relatively low selection accuracy, and at the same time, a very high selection intensity. So we would go from these 10 to 20,000 individuals here down to, we simulated 1,000, but the reality was a few hundred individuals here. So two major issues and a huge selection bottleneck from this stage to this stage. So we realized it probably would be more useful to use genomic selection already in this stage, in the seedlings. And this is how we thought this might work. We have the same structure here. We still use stage 1 and further on as our training population, because these are our clones. This is what we're interested in. But what we did now is we completely replaced phenotypic selection by genotyping our seedlings and predicted the performance of the seedlings based on the clonal information. So we're not selecting based on per se seedling performance anymore. We are selecting based on predicted performance of clones in the clonal environment. And our assumption was this would result in a massively increased selection accuracy, especially in this stage. So we can use GS to advance the seedlings and find the best clonal candidates for stage 1. GS also here, but biggest advantage, massive reduction of the generation interval. Select new parents immediately after they have been crossed. And as we always did that at the Roslin Institute, and also what we're doing now in module 2 is we use simulation, a stochastic representation of the breeding program, to figure out if our hypotheses hold true and if that works. I don't want to go into the details, but what's probably important to mention is what we simulated was a quantitative trait, or a trait that represents yield based on our assumptions of a quantitative trait. We assumed that there might be something like 20,000 QTN with really, really small effects involved in controlling the genotypes. And these were spread across 20 chromosomes. And our genetic values were modeled based on some additive and some dominance effect. So the additive effect was multiplied with some random component that kind of models genotype by year interaction. So the additive effect slightly changed from one year to the other. And the dominance component was modeled in terms of a dominance coefficient, which basically means how important is dominance compared to our additive effects. There's now a major problem, obviously. We had basically no information how important dominance is in the different traits. Dominance will have a different importance in different traits, and it will also be specific for the population we look at. So it's a highly, it's a parameter that is really, really hard to capture. So what we basically did in the simulation is we just assumed different dominance degrees from no dominance at all to almost complete dominance to cover a range what might include realistic values, and then checked what happened. And our initial approach was what probably all of us would do initially, just using the traditional or conventional genomic selection approach, where we estimate the so-called genomic estimated breeding values and then use these GEBVs to select new parents. So I'm going to show you a couple of figures which basically all looks the same and show you similar things. You have the years or the time here and some value here. What we always did is we simulated a phenotypic conventional breeding program for about 20 years, what we call the burn-in. And then at that year 0, we simulated for future 20 years all the breeding programs we compared and checked what happened. The yellow line is our breeding program where we implemented genomic selection in stage 1, and the blue line represents the breeding program where we simulated genomic selection in the seedlings already. Now, under no dominance degree, only additive effects at all. This is what you can see here. We just observe what we expected. Genomic selection strategies increase prediction accuracy, and we measured that, and we showed that this is also true in this case, and they reduce, or they help us to reduce the generation interval. And both these advantages also helped us to increase genetic gain compared to our conventional phenotypic selection breeding program. And as selection accuracy in the seedlings and also the generation interval was shorter, this breeding program outperformed this breeding program. Again, we see what we expect. Gene S was better at turning genetic variants into genetic gain. Our additive value here completely represents the genetic value because there is no dominant, there's only additive effects. And we also see that genomic selection results in a quicker increase of the inbreeding coefficient because it manages to quicker and to better increase the frequency of beneficial alleles. So it is basically what we want. We want to fixate the best alleles in our population to achieve a target individual, a target genotype. However, when we simulated different dominance degrees, this picture changed. So what you can see here, and it is somehow missing on these scales, but it is the genetic value. Here again, you can see the dominance degree 0.1, 0.3, and 0.9. The basic message is here. When the dominance degree increased, we observed that the relative performance, or that the performance of genomic selection strategies relative to the phenotypic program decreased. And in the most severe case, under a very high dominance degree of 0.9, we observed that genomic selection actually made negative gain, which is obviously not what we want, and which was at first counterintuitive because we were sure GEBV accuracy was increased and selection the generation interval was reduced. So we had the two major components tackled. But cutting a long story short, the issue we encountered was inbreeding and therefore inbreeding depression. So obviously when there is no dominance, inbreeding doesn't play a role, and we can easily tolerate high inbreeding or high inbreeding coefficients. But when dominance starts to play a role, heterozygosity plays a role, because heterozygosity leads to heterosis. And when we quickly push inbreeding, this means we lose heterozygosity, we lose heterozygous and heterotic potential, and under a high dominance degree, this becomes an issue. So a way to illustrate that is to look simultaneously at the additive value and at the dominance value in our population. Again, same concept here. We have 0.1, 0.3, and 0.9 dominance degree. And what we see in the additive value is that regardless of the dominance degree, our genomic selection strategy managed to achieve the highest rates of gain for the additive value. Genomic selection in the seedlings was always the best strategy and made the highest additive gain. However, the problem lies in the dominance value. When there is only little importance of dominance, we observe that our dominance value, due to the loss of heterozygosity, is reduced. But this is not a major problem because the increase here massively outperforms the loss. Under a dominance degree of 0.9, however, this increase that we observe is not, direction. This increase that we observe here is lower than the reduction in the dominance value that we observe here. And so we make negative gain. And a figure that summarizes a bit is something we can see here. So this is, again, genomic selection in the seedlings, 0.1 dominance degree, and our gain, our total genetic gain, the blue line, is positive and is majorly driven by our increase in the additive value, while this reduction of the dominance value slightly reduces the gain compared to the additive component. However, under a high dominance degree, this loss here is so severe that our increase in the additive value cannot compensate for that loss anymore, and we make negative gain. So we realized that probably the selection strategy of the parents is a problem here. And this is why we try to change this approach and come up with another way to select parents. And we realized that this had to be a model which includes a genomic prediction model which includes dominance or accounts for dominance. And I don't want to go into much detail with these models here, but something you would typically find in literature to include dominance effects is a model like this, where you treat your additive effects and your dominance effects both completely as random genetic effects. You have a dosage matrix which tells you 0, 1, or 2 alleles with that effect, and you have a dominance matrix which tells you if a locus either is heterozygous, it gets a 1 then, some effect, or if it is homozygous, and it will get no effect, a 0. The big problem with this equation is that random effects are drawn from a distribution where we assume the mean is 0. However, when we talk about heterosis, we assume directional heterosis. We do not assume that heterosis on average is 0, but heterosis on average has a positive effect. So a model that these authors here came up with a few years ago is a very elegant solution to that issue. And what this model basically does, it fits a covariate for homozygosity as a measure of inbreeding depression. That's what we see here. So this This parameter here describes the homozygosity, for example, percentage of homozygous loci in an individual. And we estimate a covariate of inbreeding depression, how much impact does homozygosity have on performance. And this basically is now an intercept for our dominance, which is not zero, but in terms of directional dominance will be higher than zero. And this equation helps us to get a good estimate of our additive, and if we combine these of our dominance effects. Again, another equation I don't want to go into detail with, but as we now have additive and dominance effects, what we did is we predicted the performance, the expected performance of an F1 cross between two parents. So we switched from selecting individual parents to selecting parental combinations based on the expected progeny. And when we predict the progeny, when we predict the alleles on average in the progeny, we can predict a additive and a dominance component. And we did these predictions for all possible combinations of parents and then selected the best combinations. So what we observed then is what is illustrated here in these dashed lines. You will remember the solid lines from what I've shown before, and the dashed lines are now the stage 1 GS and seedling GS strategies with parents selected based on cross prediction. And the major message here is what we observed is, regardless of the dominance degree, using cross prediction to select parents always resulted in positive gain, and it always resulted in outperforming the phenotypic breeding program, and that's exactly what we wanted. So same principle, the inbreeding coefficient, and this is the major contribution of this method. Under no dominance degree at all, again, inbreeding is kind of what we want. We want to increase the frequency of homozygous beneficial alleles. But when the dominance degree increased, our models somehow automatically compensated for that and stepwisely reduced the inbreeding coefficient. And under this very extreme scenario, what we observed is that the model even managed to maximize or to minimize inbreeding, maximize heterozygosity below the level of what we observed in the phenotypic scenario. So again, same pictures. What we see now in the dashed lines is still, when we look at the additive value and the increase of the dominance degree, our genomic selection models still manage to make good, decent, positive genetic gain for the additive value. In the dominance value, however, when dominance is somewhat important, heterozygosity is maintained over a longer period of time. If heterozygosity, if heterosis, if dominance is very important, the model even pushed heterozygosity and the heterotic potential of our population to a maximum. And the nice thing, again, is it automatically did it. It, based on the training population, found the right weights for the additive value and the dominance value without any further information coming from our side, and figured out a good balance. So coming to an end now and concluding, a conventional genomic selection approach with parents selected on genomic estimated breeding values promotes inbreeding and therefore inbreeding depression unless dominance is negligible. And this is not what we want in clonal breeding programs. So therefore, we tested genomic prediction of cross-performance, and this strategy includes dominance effects in the prediction. It always achieved the highest values, and this works because this genomic prediction of cross-performance approach the model enables an efficient simultaneous exploitation of additive and dominance effects. So this was the whole bit of a lot of complicated theory in short. I'm happy to take questions now for discussion. Just to let you know, there are a couple of questions which in general are very often pop up. And I've prepared answers in case you want to discuss them. One of them is, does that work for real polyploids? The answer is quick yes. How do clonal breeding and hybrid breeding compare with regard to that method? And there's some extensive theory. And another question is, how do genomic prediction of cross-performance and optimal contribution selection, should be OCS, not OCP, relate to each other? So these are common questions. Again, if you want to discuss one of them, I'm happy to do that. Otherwise, go on and ask me your questions. And thanks for listening.
Thanks, Christian. I think that was a great and detailed presentation. I think you made it as easy as possible to explain. But actually, I would like to first open for any clarification on the content. And I know that there was many plots, but if someone has still a doubt in some of the content, please go ahead. Otherwise, also feel free to ask a question or make a comment related to the topic. Roberto, I can see.
Yeah. Thank you. Christian, congratulations for your work and for the presentation. Could you explain again, how did you model the inbreeding effect?
So again, I don't want to go into too much detail because this gets somehow complicated. And if you want to know the details, this is explained in the AlphaSimR vignette, how that works. But just in short, we assume that we ignore epistasis because we were particularly interested in the dominance effects. And on top of that, epistasis we know is really hard to model. We don't really understand it, and we don't really know at the moment how to use it, how to specifically use it in breeding. At least that's my state of knowledge. Correct me if I'm wrong. So we assumed two components, additive and dominance. These additive effects are completely randomly drawn from a normal distribution and distributed across 20 chromosomes. So again, our assumption here, and this is the assumption that you will find in literature, probably something like 10,000, maybe 20,000, just a really high number of effects is what contributes to a quantitative trait. And because these effects are distributed across within and across chromosomes, we have two major components. One is independent recombinations between chromosomes, and we have LD and dependent recombination within chromosomes. So we think that is kind of a good representation of what we would define something like a quantitative trait. This is just some random component that we assume is some impact of G by E, some random impact of the environment, of the year effect in that case on the additive component. And we assumed diploid behavior here. Strawberries are octoploids, but they have a SNP chip with NIPs that behave like diploids. So AlphaSimR also allows to model real polyploids, allopolyploids, but in this case we assumed diploid loci with 2 alleles. And if these loci were heterozygous, we also added a dominance effect that was also drawn from a normal distribution, and the average effect of this dominance component was either 0.1, 0.3, or 0.9 of the average additive effect that was modeled.
I agree with your comment on, and maybe not going too deep on the formulas, and because I can see that some people that still have comments and questions, and maybe for the particular modeling, as you pointed out to Roberto, maybe that can be found in the papers.
I'm done anyway. But yeah, I agree. Good point. I should have cut that a bit.
No, yeah, but Christian, I think bringing this back to the practical implementation and advice and guidelines for the breeders, I guess that if I can make a question, Christian, I mean, for a long time, I guess quantitative geneticists and plant breeders were always talking about selecting based on breeding value, and you know, preaching that the dominance effects are not inherited, so therefore shouldn't be the focus of the selection. But what is exactly the change that, I mean, what changed that? Why we are saying now that we can select dominance effects, and instead of focused in the breeding value, now the focus is the genetic value. Can you elaborate a bit more on exactly what is allowing that?
Yeah, that is, I mean, that is quite complicated. I found it quite complicated to understand based on simple concepts. But what basically happens is when we estimate the genomic estimated breeding value, and if you understand the concept of the breeding value, what happens is here we model additive and dominance effects. The breeding value is based on an average effect of an allele and dominance deviation. That means the average effect of an allele already includes some dominance potential. So dominance is to some extent included in the breeding value, but the problem is the breeding value will always promote selection for one allele, because one of the alleles will have a higher effect. Dominance is included, but one of the 2 alleles will always be better. So the breeding value will promote homozygosity, regardless of inclusion of dominance effect or not. What we did now is we predicted heterozygosity in the progeny, in the offspring population. Yeah. And therefore we select for heterozygosity to some extent, or not for homozygosity. It is helpful to look at that question from the perspective of hybrid breeding, but I won't go into that if no one is interested. But maybe we have other questions.
So Christian, yeah, there are more questions. So what you're saying is that before we were selecting on the individuals that we see, so basically the dominance, we know that it was not going to be inherited, but because now we can select on the progeny and we can predict how the dominance, the combinations will appear in the progeny, somehow now we can select not only the additive component, but also the dominance component.
Exactly. You mentioned one point, there's another. So first important point is the breeding value selects individuals, and you said that, and here we select crosses. And by doing that, we also, the breeding value selects single alleles, and this selects combinations of alleles.
Yeah, I think that's definitely of practical importance for the clonally propagated crops that want to implement genomic selection and don't take it in a I guess, in the regular GS that is proposed for some line crops. And there is some questions on the chat. The first one is from Marnin. He was asking if you have thoughts or experiences with the empirical estimates of this directional dominance and how it changes over time. I guess that, yeah, I mean, I guess the question is directed to whether the dominance is going to evolve, be reduced or increase as selection occurs. Does any of the plots that you're sharing answer that question, or that needs to be investigated?
I'm not sure if the plots really show that. The first thing is, I don't really have, well, let me just comment that our results supported first question.
And on the meantime, to complement Wasim Hussain is asking, I mean, that in the clonally propagated crops, I mean, heterozygosity is being favored in nature. So the main component from his perspective, or he's also asking to the community, is that the non-additive variation, that is dominance and other things like epistasis probably, is the rule. And more important than the additive variance. Is this right?
So first thing is, I think that is not only a problem in strawberries, but in general, we don't really have estimates. And even if some studies show estimates of how important dominance is, they're based on very small samples. They're very specific for that population, and they're specific for the traits looked at. So quick answer, I don't know. I have no clue how important it is, but I think, and I'm relatively sure that there is some importance. It's not additive. Most of the, or a lot of quantitative traits, especially yield, are not only additive.
Yeah.
So a nice,
from other crops, right? I mean, maize, even being a diploid, already tell us that the non-additive variation is important, right? Is there?
Yeah. So a nice thing about this equation here, if we can do genomic selection and if we use this model, which was proposed by these authors, is we can get an estimate for how important dominance, directional dominance is. So when we estimate this inbreeding depression covariate, it will tell us how much does our performance drop when homozygosity increases by 1%, for example. And so we can get an idea if we use that model. However, I have not checked the transition of the inbreeding depression covariate over time, so I don't know how important it is over time or not. Mm-hmm. But when we go back to the three principles or the three concepts of heterosis, which are dominance, overdominance, and epistasis, if I remember correctly, we come back to the idea that we don't really observe a lot of overdominance in nature. Most of it is dominance, and this leads us to the conclusion that a line which includes all the beneficial alleles in our pool should be at least as good as a hybrid, or as good as a hybrid. So I would assume when we follow this transition over time, when we manage to recombine and set good alleles free in our pool and increase their frequency and remove the non-beneficial alleles, that over time this component of how important inbreeding or heterosis is should decrease, and our lines, like it is in wheat, where there's very little heterosis, should improve. So probably when we do that for many, many, many generations, the contribution of heterosis would be reduced.
So, Christian, we have three questions in only 10 minutes. I will go first to the one from Peter, then to Wolfgang, and then to Valeria. Peter is asking about what would be a genotyping strategy for the seedlings, I mean, in practice, to make this happen. assuming, I guess, the strawberry breeding program that you are referring to. Is there any particular recommendation on the genotyping strategy?
I'm not sure if I'm giving the answer that Peter, you're asking for. I mean, certainly because we have to genotype a huge number of seedlings here in a clonal breeding program, that is somewhat challenging. The strawberry breeders back then told us that they would be able, with the right changes to their breeding program, they would be able to do it. And I mean, just grow the seedling and take a sample.
Mm-hmm.
For genomic selection.
Would they genotype all? Would they genotype all, or just a...
Oh yeah, genotype all of them. To account for that cost, we reduced the number in the simulations of individuals. So when we started from 15,000 in the phenotypic program, we ended up with somewhat close to 11,000 genotyped individuals, but we would genotype all of them. This is the optimal scenario. If this is doable in the respective breeding program is probably another question.
Mm-hmm, mm-hmm. Definitely, that's a good question, Peter. Wolfgang, do you want to make your question or comment?
Yeah, okay. Can you hear me?
Yeah, we
I can hear you.
Okay, I think we can get quite a good idea about the importance of the dominance effect from mid-parent, mid-offspring correlation and the hybrid performance or offspring correlation with heterosis increments. And in sweet potato, in early breeding stages using 2 locations, we have for yield correlation about 0.3 for mid-parent, mid-offspring, and about 0.3 for mid-offspring heterosis increments. So my guessing would be it's about 50-50.
Mm-hmm.
And it makes a big difference when you combine these two. Also, this is so dominance is so important, and you go only for the additive effects, and you have a correlation mid-parent, mid-offspring 0.3. This is not so much.
Mm-hmm.
But when you combine this additionally with prediction of the dominance effect or you exploit heterosis increments, and there you get the 0.3 plus the 0.3, and this makes a big difference. This is very interesting, and this is I think this will be a big there will be a big change in the next years or in the next decade for clonal breeding.
Yeah. No, good.
Yeah, thanks for the comment, because it's difficult to find estimates.
Yep. And this is in potatoes, so I think it's a good also a model crop somehow in plant breeding. Christian, we have another question from Valeria. Thanks, Wolfgang. She's asking, for practical implementation of this strategy, are you suggesting that should we explore which of the 2 strategies is better, you know, the GBB or the genomic prediction of cross performance, or can we go just for one straight away?
Let me. Where the question is?
The question is from Valeria. Is on the top. I mean, she's asking in a different way in practical terms.
So
we should invest.
So it can really with a lot of confidence say.
Oh, Valeria.
Yes, Valeria.
Yeah, please go ahead.
Yes, thank you. Because as you said, Eduardo, we in several scenarios, we don't have the measurements on how much dominance over dominance epistasis effects are playing in our role. So that's the reason why. Would you suggest that we invest in take those measurements, so design specific experiments to make these measurements, and depending on how strong is the dominance, how strong are the different values, then decide whether to implement these. Or more in general, you will say, independently on the values that you have, just to be in the safe side, it's better to implement the prediction of the crosses.
Yeah, thank you. So first of all, if possible, I would suggest genomic selection in general. And if you manage to genotype and phenotype a population, the prediction eventually is just a model. So this, if the logistics are there, if you have genotypes and phenotypes, calculating the model takes a few minutes. So I mentioned that at some point, but it's good to repeat that. So when we use the cross prediction model, we get an estimate of how important heterosis is. If heterosis is not important at all, if it's completely additive, which I think is unrealistic in clones, but if it is completely additive, you can just go for the genomic estimated breeding value. However, if there is some dominance, even if it's small dominance, if it's a very low degree, if there is some dominance, the cross prediction model already starts to pay off. So again, you don't need specific experiments. You can just get your logistics or genomic selection ready, run the model, check if it induces or if it indicates, not induces, if it indicates some dominance, and if it does indicate even a low degree of dominance, use cross prediction. And the model will, as I said anyway, balance automatically the contribution of the additive and the dominance component.
Yeah.
All right, thank you.
Final question, Christian. Thanks, Valeria. There is a question from Ravi. He's talking about the fact that wheat is also a polyploid, another hexaploid. And he's wondering, I mean, is also the then prediction of a cross or genomic prediction of cross performance also important as it is in strawberry? I mean, a line crop in this case, which actually is looking to generate lines, how this modifies the picture then?
This is a complicated question. The short answer is no. There is no benefit from using cross prediction in lines, because eventually our variety will be an inbred, so only additive effects, and we can select based on GEBVs. This is really, really specific for clonal breeding programs, and also it won't contribute in hybrid breeding programs. As I said, this is a question that is often asked, but there's no extra use of this method of cross prediction in hybrid breeding programs because they work slightly differently. But in clones, where we have one population where we have to improve additive and dominance in one single step at the same time, this is where it really pays off.
Yep, and there was just a comment from Bodo about some of the logistics, cost, samples, and how in potatoes some traits are best when they are heterozygous, but other you may want homozygosity, etc., etc. But I guess what you are saying here is that the model really selects for the best of both worlds, right, Christian? I mean, it will select for the basically the alleles that should be completely additive to kind of fix them and become them homozygous, but also to keep the regions and combinations that should be in heterozygote state to keep the dominance also high.
Exactly. The model automatically figures out the importance of the relative importance of the two components and combines that in prediction. And also, Martin already answered the question, but genomic selection does not make phenotyping obsolete. Phenotyping is what drives genomic selection, and it's essential that we get abundant and good phenotypes from the clones. It makes phenotyping obsolete in the seedling stage, but this is because in this very specific scenario, our seedlings are not representative of our clonal performance. This is why we want to not select based on phenotypic performance here, but we need it in later stages. And also, very important, when we come to extremely high, or if we come to the later stages where we have many years and many replications of phenotypic performance, we would probably not use genomic selection to predict them, but we use the actual observed data. Genomic selection really pays off in early stages where there is very, very little phenotypic information.
Yeah. And just to add on top of that, as Christian is mentioning, remember, genomic selection is never about replacing phenotyping. The phenotyping strategy actually doesn't change that much in most of the cases. It's mostly about decoupling vision of product advancement and parent selection, where in the classical programs that happens at the same time. When you advance, for example, individuals from stage 1 to stage 2 is when you recycle parents, but genomic selection, what allows you is to decouple that decision. So the decision of parent selection basically happens whenever you want, at the stage that you want. And of course, that is better to do it earlier. So seedling stages, et cetera, et cetera. We got to the end of the meeting. Probably just like myself, you have other meetings starting now. So thanks, Christian, for the great presentation and the clarity answering these questions that were quite complex. And thanks to everyone for joining. Please, if you have a topic, like I said, that you want to share for the next meeting, it can be something theoretical, it can be something practical, it can be some method that you are applying, something that is working in your program. Please share it with us. So you can be only presenting very quick if you want, 10 minutes, 5, etc. We can accommodate more presenters if there is more topics. So thanks to everyone, and looking forward to hearing from you.
Yeah, thanks everyone for joining. I forgot to mention that, but there is a paper on bioRxiv. So if you want to go back and get a bit more into the details, there's something where you can read it. Thank you.
Take care, everyone. Have a great day.
Bye-bye.
Thank you. Bye.
Thank you.
Bye, Baukar. Bye-bye.
Bye-bye.
Bye-bye, everyone.
Bye.
Bye-bye. I'm gonna close it now.